Okay, so this is Chapter 3, Part 3, and our focus for this part of the chapter will be on lipids, which is our second class of macromolecules. Okay, so again, lipids are organic, large, complex macromolecules that we find inside of living organisms. A key characteristic shared by all lipids is that for the most part, for the major portion of the lipid, is going to be insoluble in water, nonpolar, and hydrophobic. Okay, and so again, I say for the most part. Most of a lipid is going to consist of just carbon and hydrogen atoms, and so again, those are going to be nonpolar covalent bonds between those atoms, and so overall, the molecule will be nonpolar, hydrophobic, insoluble in water. Although there will be a few oxygen-containing functional groups, and we will see when we get to phospholipids that phospholipids even have a special group that actually gives polarity to the molecule. And so again, in general, lipids are nonpolar. They are also the only macromolecule that's not considered a polymer. And so remember, a polymer is going to be a large complex molecule made up of repeating subunits. And so all of the other three macromolecule groups, so carbohydrates, nucleic acids, and proteins, are polymers. Lipids are the only case that are not. They are still built by dehydration synthesis, and they are still broken apart via hydrolysis. But again, they are not made of monomeric subunits. And we'll talk about what they are made of in just a few seconds. This class includes things like fats and oils. We'll look at those. It also includes phospholipids. Those are the major component of plasma membranes. We'll look at those. It also includes steroids. Okay, so steroids. We will look at cortisol and we will look at cholesterol. Okay, and lastly, we will briefly talk about waxes. Okay, so this group here, some of them will be used for energy storage, some of them will be used for structural support, and again, some of them will be key hormones, and so steroids fall into that category. Pictured here is an otter, and the otter here contains lipids, right? But specifically, I want to just talk about the lipids that are contained in this organism's fur. And so this guy will have oils and waxy substances that are going to actually kind of waterproof his coat and provide some insulation as well. And so therefore, this otter can swim in water that's pretty cold. The water's not going to get to his actual skin. And isn't going to bring his body temperature down too far, at least. Okay, we'll come back to that. Okay, so fats and oils will be our first group of lipids that we will talk about. Lipids they're common in our diet and they're important. So sometimes people will hear fats and oils and they think that that's maybe unhealthy. It really depends on what type of fat and what type of oil, right, you are consuming. So you can see in this picture here, there is some fatty fish. This is salmon, rich in omega fats, which are pretty healthy fats. We've got avocados, which again are rich in unsaturated fats. Those are good for us. Nuts and seeds. And here's some nice, maybe avocado oil or olive oil. Again, healthy fats. Fats really important in our diet. Various functions. So the maybe most important function is that it's a source of stored energy. And so excess fat in your diet is going to end up being stored in adipose cells, which are fat cells. And they're going to comprise adipose tissue, which is fat tissue. And lipids that are in there, they're being stored they're a form of long-term energy storage. Adipose tissue is also an insulator and a protector. Well, I'll come back to those in just a second. Let me just finish up on energy. And so lipids actually provide 2 times the amount of energy per gram compared to carbohydrates and proteins. So they're a really good source of energy. Okay, the preferred energy, right, in our bodies for cellular respiration is glucose. but fat can also be burned. And again, when it is broken down, it yields twice the amount of energy. So again, we store fat as an excess reserve, an excess long-term energy storage. Let me talk about insulation and protection. And so bring you back over to this picture of this polar bear here. He's got lots of insulating fat, and that's really important to his survival. That's one of the adaptations that polar organisms will have. And again, he needs that fat, right, to stay warm. So insulation. And then protection, I have a picture here of a heart. And so this heart does have some fat, okay, on the outside of it. And that fat, which is really shown here by this white substance, is really, really important in protection. And so, your heart is beating in a chamber, right? In a cavity in your body. And you want to make sure that your heart is not rubbing against other things as it's beating. So up against your sternum, right, your breastbone, or other structures that it could potentially rub up against. And so this fatty layer is important for protection. Now, too much fat around your organs, so a high amount of what we call visceral fat, is very dangerous. But a small amount is good. Okay, we got to find that balance. Not too much, not too little. Fat also plays a crucial role in the absorption of fat-soluble vitamins. So, for example, vitamin A, D, E, and K. If you're taking a vitamin, like so, for example, I take vitamin D every day, and I take that with dinner because at dinner I know that I'm going to get some healthy fats, and so I know that my vitamin D is going to get absorbed. Okay, so again, think about natural sources, foods that have these vitamins in it. You want to make sure that you're eating some fat with those foods to actually absorb those vitamins. If you don't get fat with those types of foods, then the vitamins don't end up in your bloodstream. Okay, they don't end up inside of your body. Okay, so let's take a look at triglycerol, or we could say it's a triglyceride. Really, either term is fine. This is the most abundant lipid found in living organisms. It does circulate in your blood, and it's a form of fuel, so this can be broken down for energy. Excess triglycerides are stored in adipose tissue, so in fat tissue. And again, this is our long-term energy storage molecule. Now, you don't want too much floating in your blood, right? So high levels of triglycerides can be dangerous, but you do need some for energy. Okay? Okay, so let's look at triglycerides. You're going to actually build one of these in lab, and you're going to have to sketch one in lecture, and so you want to make sure that you understand the structure. So this is what it's made of. This is the finished product down here. Okay, so what's it made of? It's made up of a molecule of glycerol, and glycerol contains 3 carbons, and each carbon is bonded to a hydroxyl group. And then the other positions on carbon are filled by hydrogen atoms. So this is glycerol. And then there are 3 fatty acids. So if I come down here to my final molecule, right, this is glycerol, carbon 1, 2, and 3. And then these things are 3 different fatty acids. And so this is what a fatty acid looks like. So there's a long hydrocarbon. And then right here at the end is a carboxyl group. Okay. In lecture, you're going to be drawing, and in lab, building a fatty acid that's much shorter than this. But in nature, fatty acids tend to be very long. Okay. And so the finished product here, triglycerol, is the result of this fatty acid chain getting attached to this glycerol. And so the attachment's going to happen at these hydroxyl groups. So one fatty acid is going to go here, the second one's going to go here, and the third one's going to go here. And as that happens, it's dehydration synthesis, so we're going to pull out water. And so circled in this kind of reddish color shows you for every fatty acid that gets attached, here's an OH group, and here's a hydrogen that comes off. So for every fatty acid that gets attached, we pull out 1 molecule of water. And so in this final product, that happened 3 times. So once, twice, and 3 times. Okay, so the bond that forms here, we're going to give it a special name. We're going to call it an ester linkage. So there's 1, 2, 3 ester linkages here. Okay, remember in carbohydrates, we called the bond between 2 monomers, we called it a glycosidic linkage or a glycosidic bond. And now in lipids, we give it a special name. We call it an ester bond. Okay? So that's the covalent bond in between the glycerol and the fatty acid. Okay, so you've probably heard of saturated and unsaturated fats, and so I want to make sure that we understand what those are. So saturated fats are going to be fats in which, if we look at the fatty acids, there's going to be all single bonds between the carbon atoms. Okay, so butter is a great example of a saturated fat. Here are the triglycerides of butter. And so this looks just like the molecule on the other page, right? This was glycerol, and then there's 1, 2, 3 fatty acids. And so this is a skeleton drawing. So at each point here, you have to assume, you just have to know, that there's a carbon bonded to 2 hydrogens. And you can see between all of these carbons here, here, and here, there's no double bonds. They're all single bonds between the carbon. Okay, and so you end up with fatty acids that are pretty straight, pretty linear, and they can stack up on top of each other. There's no kinks or bends, and so you end up with a pretty tightly packed molecule. And so therefore, saturated fats tend to be solid at room temperature because these fatty acids compact so tightly together. We find saturated fats in animal products like meat, beef, poultry, and dairy products. And saturated fats, consuming excessive amounts of saturated fats has been linked to an increased risk of cardiovascular disease. And so you want to limit your fatty acids in your diet. You don't need to eliminate them, but you should limit them. Okay, unsaturated fats, on the other hand, they're going to have at least one double bond in the fatty acid tails. And so let's look here at the structure. And so this is a saturated fatty acid tail. Here's a saturated fatty acid, but right here, this is unsaturated. And because of this double bond, this bends or kinks, and you can see that structurally here. Okay, and because of that, this is not going to be so compacted as we saw in butter and saturated fats. There's space in between the molecules. Now, if there's just one double bond, we call that a monounsaturated fat. Multiple double bonds, so even more kinks, would be a polyunsaturated fat. Okay, so again, these double bonds that we see here, they end up kinking the molecule, making them less compact, and we say that that fat is more fluid, and so they tend to be liquid at room temperature. So in our example here, this is a nice healthy fat. This is olive oil. Unsaturated fats are found in plant-based foods like vegetable oils, nuts, seeds, and avocados. Okay, and consuming these still in moderation, right? You also don't want these in excess because fat gets stored, right? And so consuming these in moderation can actually have really positive health benefits, such as a reduced risk of cardiovascular disease. Okay, and so we're going to move to trans fats, and so you've probably also heard of these. And so just to remind you, this first example, this is stearic acid. This is a fatty acid that doesn't have any double bonds. So if this was the 3 components of my triglyceride, that would be a saturated fat. Okay, now here's an unsaturated fat. Here's the double bond. Look at that nice kink. Okay, we say that this molecule is in the cis conformation. So what does that mean? Cis means same side. If you can remember back to when we talked about isomers in the last chapter, this is in the cis configuration. Okay, so the hydrogens here are on the same side of the double bond. The opposite of that is a trans fat. And so in a trans fat, again, there's a double bond, but look at the hydrogen positions. They're on opposite sides of the double bond. And so unfortunately, because of that, this molecule tends to be straight. Even though it has a double bond, it's not kinked. So a trans fat really looks like a saturated fat, and it behaves like a saturated fat. Okay? Trans fats are found in processed foods. We actually change the fat to the trans configuration. So processed foods like fried foods, baked goods, margarine, and snack foods. Trans fats are very, very low. So I don't want to say they're not ever found in natural food sources, because they are, but very limited amounts. So usually what we actually force trans fats. We chemically modify healthier fats through a process called hydrogenation. And what that is we add hydrogen atoms to liquid vegetable oils, so changing them and making them more solid at room temperature. And we do that to increase the shelf life. But that's not healthy. They're very bad. And so there's actually been lots of studies done to show how bad they are. They raise our LDL, low density lipoprotein, which is your bad cholesterol, and then they lower your good cholesterol, which is called your HDL. And so a diet high in trans fats will increase the risk of heart disease, stroke, type 2 diabetes, so on and so forth. They're very, very bad. They're so bad that in 2018, the FDA recognized this, and they marked them not safe for consumption, and they have actually forced manufacturers to remove trans fats from their products. Okay, so they're banned. So that's a good thing. Okay, the last fat that I want to just talk about are omega fatty acids. And so you've probably heard of these as well. These are essential polyunsaturated fatty acids. So they have multiple double bonds. And that term essential means that our bodies cannot make these. And so we have to get these from our diet. So it's important to eat foods that are rich in this polyunsaturated fat. There is a certain ratio that you should get of omega-3 to omega-6. And that is important, but in general, omega fats are good for us. And so here we go. So if this is the fatty acid, this is that carboxyl end, right? And so here's 1, 2, 3 double bonds. And because of that, look at this. It gets this nice round shape. Lots of kinks in this molecule. And so this, again, is going to be at liquid at room temperature. Eating foods that are rich in polyunsaturated fats is good. It reduces your risk of heart attack. It lowers triglyceride levels in the blood, decreases blood pressure, prevents blood clots, reduces inflammation, reduces your risk of cancer. They're very healthy. Okay. Polyunsaturated healthy fats. I'm going to quickly talk about waxes. I only have one slide on this. So I mentioned this at the very beginning of this part of the chapter. There's our otter again, the same picture. So waxes are going to be lipids. They're going to be solid at room temperature. They are made of a little bit different than a triglyceride. So they're made of long-chain fatty acids, like we've already seen, but those fatty acids will combine with either alcohols or other hydrocarbon chains. So they have a different structure. They're really important. They provide a protective coating on surfaces of plants, animals, and insects. And so in this particular example here, I'll talk about the leaves first, which are our lower picture. Leaves, and you're actually, if you take General Biology 2 with us, you'll see slides of leaves, and you'll actually see the wax, which is called cutin. We'll talk about the function there as well, but just a quick little overview of their function. This wax is going to be produced. It's going to provide a protective coating that's going to reduce water loss from the plant. And so evaporation is happening and plants could dry out pretty quickly. So this waxy layer is going to prevent water loss, and it also provides protection against pests and pathogens. So it's a nice waxy coating on the leaf. And animals, I already mentioned this, like the otter and other aquatic birds, and on those aquatic birds, on their feathers, they're going to have this waxy substance that's going to, again, provide kind of a waterproofing. Okay, so that's wax. Wax also has industrial uses, right? We make candles out of it, and other things. Okay. Okay, so we're going to talk about phospholipids, and we're getting close to being done. So phospholipids, this is a really important class of lipids. We are going to come back to this in both chapters 4 and then again in 5. And chapter 5 focuses on the membrane, and so we really talk about phospholipids again. Just want to introduce you to them now. So when you hear phospholipids, I want you to think cell membrane. And again, we haven't looked really closely at cell structures yet, but the membrane is going to be the outermost barrier in animal cells. And in plants also have them, but they do have a cell wall outside of the membrane. In both plants and animals, all organisms, the membrane is extremely important. So again, whether we're looking at bacteria or plants or fungi or protists or animals, we all have the same basic membrane structure. And so phospholipids are going to be the major molecule making up plasma membranes. We use the term, oh wait, before I get there, let me just show you its structure. So it's made of glycerol, which you've seen, 2 fatty acid chains, which we've seen, right? We saw both of those when we looked at triglycerides, but here's the different part. It also contains a phosphate group. So instead of having 3 fatty acid chains, it has 2 plus a phosphate group. So let's look. Here's glycerol, 1, 2, 3 carbons long, and glycerol is going to be attached to 1, 2 fatty acids. This one's saturated, this one's unsaturated. And then on that last carbon, instead of having a third fatty acid like we would see in a triglyceride, instead it has this group. This is called a phosphate group. So in the very middle of that, that's phosphorus attached to 4 oxygens. So this is a phosphate group. Look at the charge on the oxygen. That's really important. Okay, so let's get down. Let's go down here. I want to talk about this term amphipathic. You're going to see this. So amphipathic is going to be, it means that this molecule has 2 distinct regions that are going to behave differently. They're chemically different and they're going to behave differently. And so when we look at this phospholipid, it has one end that is hydrophilic, water-loving, wants to associate with water, and one end that is hydrophobic, water-fearing, doesn't want to associate with water. And so this region right here, which we are going to call the polar hydrophilic head group, is going to want to associate with water. And that's all because of this charge, right? We've got this negative charge here, so this becomes polar hydrophilic. Where these tails, where the fatty acids are, we say that these are hydrophobic tails, water-fearing. Again, amphipathic, all one molecule, but 2 very distinct, very different regions. Okay, that's important to the membrane structure, which I'm going to show you here. So because of those properties, because phospholipids are amphipathic and they have separate regions that behave different, they're going to form a phospholipid bilayer. So if I literally just took a handful of phospholipids and just threw them into water, this is what would happen. They would arrange themselves this way, just based on hydrophobic and hydrophilic interactions. Okay, and so you can see all these red spheres here and here. Those are those hydrophobic heads. And you can see that the tails, the hydrophobic tails, are facing each other. So this is called a lipid bilayer, a phospholipid bilayer. And so if this was a cell, this would be outside the cell. Okay, and outside the cell is usually an aqueous, watery environment. And this would be the other side of the membrane, which would be in the cell. This would be the cytoplasm. Again, an aqueous, watery environment. And so those polar head groups like water. So they're going to face the water. And these tails are hydrophobic. They hate water. And so they're going to face inward and not let any water in here. And not let really anything else that's polar or charged or hydrophilic in here. And so it creates a really neat structure and really neat selective permeability. So that's right here. So again, this phospholipid bilayer is going to be a barrier. It's going to control what comes in and what goes out of the cell. So it allows for what we call selective permeability. We say that the membrane is semi-permeable. It's going to let some stuff cross in or out, but other stuff can't without help of another molecule. And we'll talk about that actually soon in the next part of the chapter when we talk about proteins. Okay, so membrane structure is going to be really, really important. Last slide. The last type of lipid that we'll talk about are this class called steroids. And so steroids, again, are going to be hydrophobic, insoluble in water, just like all of our other phospholipids, or sorry, all of our other lipid molecules except for phospholipids are a little bit different. They do have a polar region. Steroids are going to be grouped together because they have 4 linked carbon rings. And so this is an example here that cholesterol is an example of a steroid, and so is cortisol. You may have heard of cortisol. Cortisol is a stress hormone. Okay, it is a lipid. So some hormones are proteins and then some are lipids. This is an example of a lipid hormone. So you can see 1, 2, 3, 4 rings. 1, 2, 3, 4 rings. Okay, so they have 4 linked carbon rings. Cholesterol is going to be the most common steroid. This is made in our liver, and it's a precursor to many hormones like testosterone, estradiol, and vitamin D. Those are all lipid-based hormones. So it's made in the liver, but we also need to consume a little bit of cholesterol in our diet. But we do make cholesterol. Cholesterol is also a component in animal cell membranes. And again, when we get into chapter 5, when we really look at the membrane, we'll talk more about cholesterol. But just to let you know, cholesterol, because it's hydrophobic, it will basically be embedded in the lipid bilayer. Okay, and it's going to be what we call a fluidity buffer. It helps the membrane maintain really its shape, its fluidity, even if temperature is changing. So cholesterol is really important for homeostasis. And that is the end of this part of the chapter.